and thank you. I would like first to thank the organizer of ParAqua having me here today. It's a very nice experience indeed. So my idea is that I would like to present you what we study within my equipe in France at the IRA Institute of Social Antipolis. And we are mostly, I mean, me in my project, but also the equipe, we are more focused on the understanding of the infection of Phytophthora on plants, so on the lyric plant. And what is interesting for us is to understand the mechanism of motion, which are adopted by the to swim toward the plant and start the infection. So let me present you a little bit these aspects. So the mechanism of motion in swimming eukaryotes. So we are going to develop a little bit this point. And the idea is that to provide you a little bit of basic knowledge on this aspect that you may be used after to develop tools for the identification and detection of your organism based on the motion mechanism. And we have two sessions. A first session, which is this more theoretical one, and the second session, which is more practical, but indeed is a kind of demonstration on how to use software for image analysis to again characterize the motion of your organism, but it's a demonstration. We will provide you a movie that if you want later for the one that you're interested, we can analyze together after the class, because otherwise it takes a lot of time. Okay, let's get it. So a little bit of incipit presentation. So I'm a presentation student and I'm at the Institute Sofia Global. At the Institute, we are part of CNRS in France or IRA, and we are also the University of Strasbourg. And we are mainly focused on the plant biotic interaction. My equipe of research is IPO, interaction plantometry. So we are particularly focused on the interaction between the plants and the mycel, and particularly between plants and Phytophthora. So we can. Okay, so briefly, what we will see together in this first hour of class, I hope. So we will see a little bit in general what is cell migration. We will see more in detail the different migration mechanisms that our microorganisms acquire during evolution. We are going to see in detail the structure of the flagella in eukaryotes and eukaryotes. A little bit, we are going to understand how also works the sensing mechanism in the microorganism. And finally, I would like to focus a little bit more attention on my pathogenic model, which again is represented by Phytophthora spore. And within this context, I would like to discuss a little bit with you about the pathogenic model interaction that we have between pathogen and plants, because I think it's interesting for me at least to have a discussion with you if it's the same one in the case of aquatic planted. So it's to see how the plant responds to pathogen and plants. And finally, I would like to present you some microscopic devices which we currently use in our lab to analyze the motion of our zoospore. Again, it's a theory, more theoretical, but later on we try to give more practical input on this point. Okay. So, the color completely changed. Yeah, no problem. Okay, so cell migration, as you may know, is a fundamental biological process. If we think to multicellular and complex organisms, it's a fundamental process for biological functions such as embryogenesis, immune defense, or reparation mechanism. When we pass to smaller, more simple cellular organisms, is again a very biological function, which is essential for the colonization of the biosphere. For example, if we think about our microorganism, the cell migration is important for them to search for food, to search for energy, so to colonize the biosphere and complete their life cycle. So, as you may know, cell migration is a property that is shared between basically all living organisms. So all living organisms in the heart, in our stomach, sorry, can actively move, excluded the plants that for the moment go to the other side. And also from it's also valid for most of the food, like most of the food, as we see, crickets, for example, are projects that have developed way of motion thanks to their sport form. Okay, so let's say that all living organisms are able to move and that they are able to move thanks to different mechanisms. So we can go from the smallest flagella, which are present on the bacteria, to the quite big fin of a 10 meters away. So very different mechanisms. Very in general, just to give you an idea of the different self-regulation mechanisms. In macroorganisms, we can have terrestrial locomotion mechanism, which include, for example, walking and running. As you might know, walking is a locomotion which is characterized by the fact that we have 4 points with 1 foot, 1 leg on the ground, while running is characterized on the other side by an aerial phase. So during the running, we have this kind of very romantic phase in which we are fluctuating. Yes. But of course, microorganisms also have developed some of them, not only terrestrial locomotion, but also aquatic, aerial locomotion, for example, flying, which is sustained by the flapping of the wings, or also swimming, which is an aquatic way of locomotion, which can be proposed by different systems. Now, what is interesting for us, of course, is not to understand how a rhino works, but indeed it's more interesting for us to evaluate the mechanism of migration in microorganisms. Again, in microorganisms, we have many different ways of motion. One of them is for sure swimming, but this time the swimming of the bacteria is quite different from the swimming of the whale. In this case, the swimming is defined as the individual cell movement in liquid or low viscosity conditions powered by flagellar rotation. So we are, in this case, in liquid and in low viscosity condition. And we have a flagella, which is the motor mechanism of the movement. But we can also have other kind of mechanisms, such as the swarming, which in this case is defined as the rapid multicellular movement across surfaces powered by flagellar rotation. So basically in swimming, we have one cell which is moving to liquid and flagella, and in swarming, we have multiple cells that are moving together on surfaces by flagella. But we can also have, for example, gliding, which is defined as the active movement on surface powered by photon adhesion surfaces. So many different mechanisms, and I just want to give you an idea of what we can find. Once again, I think that the interest, not of everybody, I would say, but of the majority, is to a little bit understand this kind of dynamics, but in the context of a microorganism. When we speak about microorganisms, we can make a main distinction between bacteria, which are characterized by small size, including between 1 micrometer to a few micrometers, and protozoa, which are characterized this time by higher size. So including between 10 micrometers and 2 millimeters. So anyway, both in the case of bacteria and protozoa, we are of course in a micrometer scale. We are in a micrometer world, and when we are in this kind of world, very, very, very small world, we have to consider that the physical laws that govern this world are quite different from those that are, let's say, that govern our macrometer world, macrometer scale. And some of them we will see has to take into account, and normally are laws that, for example, do not really valid in our scale. I would like to detail now this physical aspect. It will be hard, but you tell me if you cannot understand. So basically, what does it mean moving in a micrometer scale. So again, moving in a micrometer scale means that we have to take into account physical constraints that normally are neglected in our condition. In our condition, that in this case, and all the physics present will detect me, but I will call it normal scale world, or normal size world. Sorry, in our normal scale world, normal scale size, are through particular laws. So, for example, the second law of Newton. According to this law, we have that the force which is applied to an object is the result of the mass of the object and of its acceleration. And in our again word scale size is also true. It's also valid. The inertial property, according to which basically an object is able to maintain the movement without that the force is applied. So this is because of inertia. To give you a more concrete example, imagine yourself that you hit a ball. The ball will continue to move and fluctuate into the air, not because of a force, because you apply the initial force, but because of inertia. So in our world, inertia helps a lot because it allows us not to use always the force. Okay. This is not very true. It's a little bit different when we are in a micrometer scale. When we are in a micrometer scale, we are in a different environment. In this environment, we don't have any more the inertia, but we have other forces, which are called viscous forces. Those viscous forces dominate over inertia. And this means that a microorganism, to move in this condition, always has to apply a lot of force. No inertia that helps them. Okay. To be more concrete, what does it mean to move in a no inertia condition, and where you have viscous forces that dominate over inertia? More or less is the case of you swimming in a pool of waves. So, as you can imagine, the movement in a pool of molasses, where it is a very viscous environment, it would be very complicated, and you would probably have a lot of struggling in movement by using your normal motion mechanism, and probably you will not move too. So on the other side, in the permanent and the evolution, were able to develop mechanisms that enable them to overcome these viscous forces, and to efficiently move in such an environment. As we can see here in the case of our sport, which can reach high speed velocity and move for long distances. Okay, is it a little bit clear? To go a little bit more in detail, as I told you, microorganisms were able, thanks to evolution, to evolve a particular swimming mechanism. And in the case of prokaryotes, we speak about the bacteria flagella. You probably may know the bacteria flagella, but quickly, the flagella is a complex machinery which is highly conserved among bacteria and which is mainly composed in three modules. We have a basal body, which represents the rotary motor of the flagella and which is embedded in the cell wall of the bacteria. The basal body is made of different rings, and within these rings module, we find an internal rod which is able to rotate. We have then the hook, which is a curved protein, which is attached to the rod within the rings of the basal body and attached in the external part to the filament of the flagella. Finally, we have our filament, which is basically made of repeating the unit of cytokine, and which can have a different length according to the species of the bacteria. How does it work? So basically the principle is that flagella rotate, and the rotation of the flagella is activated by an electrical chemical gradient which is generated on the cell wall of the bacteria. Basically, we have a flow of hydrogen and sodium ions which go across the cell membrane, and this flow will enable the rod within the rings of the basal body to rotate. The rotation of the rod will then enable again the rotation of the filament of the flagella. Finally, this rotation is very efficient because it can allow the prokaryotic organism to move and also to reach a high frequency in the rotation of the flagella. Okay. So prokaryotes, what happens in eukaryotes? This story is a little bit different in eukaryotes. Indeed, in eukaryotes, we speak about eukaryotic cilia or flagella. Do you know the difference between the bacteria flagella and eukaryotic flagella? Yes? No. Okay. Yes. So the eukaryotic cilia or flagella basically is an extension of the body of the eukaryotic microorganism, of the membrane of the eukaryotic microorganism. So it's not something which is anchored like in the case of bacteria, but it's really an extension which protuberates from the body of the organism itself. Again, cilia and flagella are very well conserved among eukaryotes, so you can have a very different way of motion of flagella, but the structure is almost always the same. So the flagella and the cilia or flagella, it's the same, we will see later the differences, basically originate from a triplet of a centriole, which are also called basal body of the cilia and flagella. From this basal body of the cilia and flagella, we have the development of the central axon. This axon, as we can see here in the cross section, is basically made of nine microtubules, 9 lateral microtubules, and the central pair of microtubules. Between the different microtubules, we can find motor proteins such as the main arms, but also connecting proteins such as nexin or radial spoke. So the nexin and the radial spoke. Again, how does it work this time? So in this case, the mechanism of motion is a little bit different, and I would not say that it is not really understood, but still there are different models that have been proposed to explain how the flagella and the cilia in this case enable the movement. Among the first models that have been proposed, we find the bending models. The first bending model is the contraction bending models. In this case, we have that the flagella bend, okay, and the bending of the flagella is caused by contraction. And contraction is caused by the fact that each filament is able to short on one side, and it's called the contraction. Another bending model proposed wants that DNA proteins, that as we can see are the motor proteins that connect the different pairs of microtubules, use ATP energy to provoke the microtubule sliding. So you have to imagine two microtubules that do that. And because microtubules are interconnected at the base of the axon, we will have that this sliding of microtubules on each other is proposed to the other pairs of microtubules. Finally, all the pairs of microtubules will slide and will enable the flagella to bend. A more complicated model is the beating model. In this case, basically we have that the flagella is beating over time. So it's acquiring a particular oscillation pattern over the time. In the case of the beating model or mechanical feedback model, we have to imagine our axon divided in two sections. And in these two sections, we have two exact sections of linear arms, which pull one on the other side. So this is the axonium, these are the two sections of linear that do this kind of movement. Basically, the beating of the flagellum will be the result of the winner section of linear arms, which pull along with the other section of the linear arm, which are the loser ones. So kind of like this. And the beating is generated by the elastic forces, which are generated again by this kind of movement, this kind of pulling between the two sides of the linear arms. Of course, when at the two sides of the axon, the linear pull with the same intensity, the result of the force will be zero. So we will not have beating at all. So no oscillation of flagella over time. Is it okay? So as promised below, is very frequently to understand the difference between cilia and flagella. You might know that, but basically the main difference between flagellate and ciliate is that flagellate are characterized by a small number of long flagella distributed on the cell body. And the main function of the flagella in this case is the propulsion force to help the organism to move. While in the case of ciliate, we have a high number of short or more short cilia, which cover the cell body, and then of course help this organism to move, but also can do very different other function. For example, the transport of the surrounding fluids and elements. Think about maybe something which is more close to your area, that use the cilia to transport the bacteria nutrient within himself to in order, this work can be based on the different scale of velocities. Indeed, this very nice publication from 2018 highlighted how basically the ciliate are able to span on a larger range of speed compared to flagellate. And the ability of ciliate maybe to move faster is also due to the fact that they have more that maybe propel them a little bit more compared to the flagellate. Okay, just to understand the differences. So now what we thought was interesting to a little bit evaluate was how at one point during the evolution, the motion mechanism, so all these mechanisms that we have seen that have been developed thanks to the development of structure for motion, and the coupling of this motion element with the sensing ability of microorganism represent a crucial point for the efficient colonization of the biosphere. So, very in general, we have progressed from a passive way of movement, which here in this case is represented by the Brownian motion. In the Brownian motion, we have basically particles which passively move because are pushed by other small particles. So we don't have any mechanism of motion, just random and passive movement. But from that, we have seen that we were able to go through an active way of movement, which is propelled by the Carlotta flagella or prokaryotic bacteria. So I would say a good point for our microorganism. But again, from that, our microorganisms were able to detect not only a system that allowed them to run straight in one direction, but also develop the ability to change the direction stochastically. And we will see how. This mechanism of running in one direction and then change in the direction in prokaryotes is called run and tumble motility. Do you know run and tumble? So during run and tumble motility, we basically have two different phases, run and tumble. During the run phase, we have that the flagella of the bacteria form a bundle, and the bacteria moves forward in one direction. When the flagella are in this bundle, they move during the run phase with a counterclockwise rotation. At one point, stochastically, so randomly, they turn. And the turning phase is called tumbling. During the tumbling phase, we have that one or few flagella, as you can see here from the image. They basically exit from the previously formed bundle, and they reverse their rotational direction, and basically allow the bacteria, the bacteria for this change of direction and to move in another direction. In this case, the flagella will move with a clockwise rotation. After the tumble phase, we will have a run phase in a particular direction with the same properties of before. This run and tumble allows the organism to move around, but also to go in the direction or away of a particular stimuli that we will see after, which kind of stimuli they can sense. So they can move towards to or away from. In eukaryotes, again, this mechanism are a little bit more complex, I would say, and really specific to the different species. We don't have a true run and tumble motility in eukaryotes, but sometimes we can have some kind of mechanisms that resemble it. In this particular case, I want to present you the motility pattern in phytophthora spores, which are the organisms that I study, so it's not just me. And that also I found it interesting because it's very different, but a little bit resembles the run and tumble prokaryote. In this case, we have our spores, which perform straight run. On the straight run of this spore, we have the two opposite flagella, which move with the coordinated finger. Then, at one point, our spore can change their direction, so you kind of a tumble phase. And in this case, when they change the direction, they stop to beat the posterior flagellum and they continue to move with the anterior flagellum, looking for a new direction. Again, after this kind of tumbling event, we have a run phase, where again we have the two anterior and posterior flagella, which move and in a coordinated way. So this is a typical mechanism that we can find in our spore, and most likely we can find in other eukaryotes which have two different flagella. But for example, we already know that it's not a mechanism that we can find in eukaryotes, that they have two similar flagella, identical flagella, like for example, Chlamydomonas. Okay, as I told you, our organisms are able to run, to change direction stochastically, but they are also able during the run phase to sense different stimuli. Which kind of stimuli? Of course, we can have thousands of stimuli, but just to give you an example, we can have, for example, chemical stimuli or electrical stimuli. Of course, there are just an example. We can have mechanical stimuli, photo stimuli, but just to illustrate a little bit the principle. In the case of the chemical stimuli, we normally have a molecule which originates in the organism, a response in the terms of movement. The response in terms of movement in the case of the chemical stimuli is called chemotaxis. So, for example, the organism can sense the presence of a particular molecule, let's say glucose, and we can say, I'm interested in it, so I go toward the glucose, I take it away from glucose. And this particular response in terms of taxis is called chemotaxis. Something similar can occur in electrical stimuli, where this time we don't have a chemical molecule, but we have an electrical stimuli. And the taxis, so the response in terms of movement to this stimuli is called galvanotaxis. I hope you don't. Okay, I will move on. Okay, so fantastic. Our organisms are able to sense a particular stimuli, and they're able to respond to the stimuli. So they have developed sensing mechanism. We have two kind of different sensing mechanisms in our organism. We can have temporal detection, so a time detection, and a spatial detection mechanism. The temporal detection mechanism is something which is more related to small size cells. So, for example, to our bacteria. And in this case, very general, we have that our cell of a prokaryotic organism, for example, bacteria, is able to sense a particular signal through a receptor. The receptor, the signal, is transduced in a transducing network, and the result of the transduced network of the signal will be a mechanical response, which means that the flagella will change the rotation. So probably the organism will change the direction, we can assume, but also the adaptation mechanism. Adaptation mechanism, it means that basically the bacteria, the small size cell, thanks to the mechanism, will be able in the future to not only respond to the same stimuli, but to respond to gradient of the same stimuli, which is something very important. The different mechanism is the spatial detection. The spatial detection, on the opposite, is something that which is more related to big size cells. So let's say eukaryotes. In this case, we have again the sensing of a particular stimuli through a receptor, two different receptors which are localized in different parts of the cell, because in this case we have a bigger cell. We have again the transduction of the signal, and this time the transduction of the signal will result in a mechanical response, because again we have a change in the flagella behavior or in the motion of the organism, but it will also result in the cell polarization. And why cell polarization? Because again, we are dealing with big cell, with polarization is possible, while the smaller cell is not really possible. It's a little bit complicated, but keep in mind that it's partial detection versus cell detection. Okay, finally, as I told you, I would like now to a little bit focus more attention on the motion mechanism and infection mechanism which are adopted by the organisms that I have studied, which is Phytophthora. In these three of unicellular swimming eukaryotes, we can find many different organisms, and among them we can also find parasitic organisms. Now, what I'm interested in are zoosporic parasitic organisms, which in the case of Phytophthora can be grouped under the staminophyll domain. In this case, we cannot see Phytophthora because Phytophthora is characterized by a stage which is multicellular and a unicellular stage. So it's not appearing in the graph. And if I say something wrong, you will correct me. Okay, so Phytophthora is a telluric plant pathogen. It is a filamentous microorganism producing biflagellated zoospore during asexual reproduction. which enable the organism to explore the soil particle. It's very interesting Phytophthora in the agricultural, but I would say not only agricultural, also environmental field, because it's a pathogen which causes millions of agronomic losses in agriculture, but it also attacks the natural environment. The problem with Phytophthora is that it is also quite difficult to control in the field because it has developed a resistance to the chemical treatment, and most of the chemical treatment that are used against the fungi normally do not work on mice because of the morphological differences that we have analyzed in this. So, as I told you, infectious agents of Phytophthora represented by zoospore, which are produced during asexual reproduction. The main characteristic of the zoospore of our Phytophthora is that they have two morphological different flagella. An anterior flagellum, which is decorated by tripartite tubular hair-like structure, the mastigonium. That is the reason why I guess they are part of this Staphylococcus. While the posterior flagellum, which is a posterior smooth flagellum, which, as you can see, does not present this mastigonium structures. The zoospores are able to respond to different stimuli and, of course, are able to detect a plant in the soil, so to sense signals coming from the plant and go toward the direction of the plant to start the infection. The mechanism of the zoospore movement, the movement mechanism of the spore, we kind of deal with them before, but again, basically we have the two flagella will be longitudinally along the anterior and posterior axis and enable the zoospore to reach high speed velocities. And basically, as I showed you before, we have that during the downward event, the posterior flagella stop to beat, while the anterior flagella, which is the one decorated with the mastigonial hair, continue to move, kind of redirecting the direction of the zoospore. So now, zoospores are able to detect the plant and are able to swim in the soil toward the plant root. When they arrive on the plant root surface, they initiate the germination by insistence. So we have that the zoospore arrive, the zoospore will insist, meaning that it will generate a cell wall and it will generate in a crystalline-like structure, which will penetrate the root surface and which enable further colonization of the plant. Now, we have to know, and I will try to be synthetic on this point, that the different phases of Phytophthora plant interaction follow the model, which is basically the one recognized between plant pathologies, which is called the deep-dive model, or PTI-ATS API paradigm. Do you know it? Okay. So, in this model, zigzag model, which has been proposed by Dander in 2006, so we have basically four different phases. The first phase, where the plants are able to recognize pump, which are also called pathogen-associated molecular patterns by particular receptor. The recognition of the pump in the plant will result in the pump-triggered immunity. Immunity that the plant uses to halt the further colonization of the microorganisms of the pathogen. But successful pathogens during the evolution, they have developed effectors which are able to interfere with the PTI, with the defense of the plant, and trigger susceptibility in the plant. This susceptibility triggering the plant will now enable our pathogen to take nutrition from the plant and in general attack it and take advantage from it. But again, evolution not only help the bad guys, but also the good one in the plants. Because during evolution, plants were also able to recognize one specific effect developed by in this one red. And this particular recognition, which is a very specific recognition between a plant, a particular strain of pathogen, linked after the API, which is the effector triggered immunity, which consists basically in an amplified API response, which results disease resistance and above a particular threshold to the hypersensitive response. The hypersensitive response is a response that plants use basically to delimitate the area of infection on a particular tissue. So basically what the plants do is to start a program of cell death to delimitate the area of infection and basically to stop the spread of the pathogen. Finally, we have the work phase. Again, during the evolution, selected pathogens were able to gain new effectors which help the pathogen to suppress the API of the plant and to make the plant susceptible again. But again, selected plants develop new MDLR for the recognition of new effectors, which result again in the API. So as you can see, the zigzag model that can be, let's say, productive over the time. So here is just very quickly an example of how some of these, let's say, actors that play a role during the plant pathogen, during the infection of the plant, so in the zigzag model, are also strictly related to motility. For example, we have that flagellin, which is the main component of the bacteria flagella, is involved in this zigzag model since it can be a pump, so it can provoke the PTI response. And what is interesting for us is to understand if we have a similar molecule, similar structure in my Phytophthora zoospore flagellum that are also involved in this dialogue with the plant, this dialogue during the infection of the plant. For the moment, I don't know, but since I'm doing some preclinical analysis of the flagella, I hope to get some insights on this point. Okay, that's the finish because I have 9 minutes. As I told you, now I would like a little bit to give you more theoretical insights on microfluidic approaches for the analysis of zoosporic pathogen emission. Later, we will develop it more on a practical way. So, in general, microfluidic is defined as both the science, which studies the behavior of fluid through microchannel, and the technology of manufacturing basically miniaturized circuits where we have miniaturized channel and miniaturized chamber where we can confine a particular object that we want to analyze and control the condition of the reading. Microfluidic devices are important technology nowadays in many fields of biology, from cell biology to disease diagnostic, since they enable a high spatial and temporal resolution. So you can really control the different conditions in your device and you can make quite good observations. In particular, in our lab, we are using and we have already used microfluidic devices for this analysis of the measurements we can box. That is the response of it. Microorganism to particular organism. So again, this kind of approach enable inaccurate control over the channel because it's true that you can decide how you want your channel, how high or high you want your channel, and also enable a control over the chemical molecule that you want to test on the microorganism. So you really have a control of the whole condition. Also, we can use often for this microfluidic device some kind of polymers like PDMS, which are transparent, which are easy to manipulate and low toxic, which enable you really a good visualization of the response of your cell to a particular molecule or a particular sequence. And apart from that, you have a control. So on the general condition, you have the control on the organism that you put inside the material channel, and you also have the control on the molecule that you insert to test on your particular object. This is an example. This is a PDMS microfluidic circuit that we have used to analyze zoospore subjected to a flow of a potassium gradient. As you can see here, in this circuit, we have a basic channel, which are inserted as cross. We have basically a central channel where we inject the spore, and lateral channel where we can inject potassium solution. Finally, we are able to analyze the response of our spore here in the hemostatic chamber, where our spore at this point will be completely subjected to the gradient that we have created with the potassium. Okay, so this is just an example. Now, what is interesting to analyze when we are in this condition is, for example, analyze how the zoospore or our organism distribute in response to a particular molecule. Or also, what is interesting to analyze is how and if, for example, the velocity of our zoospore in response to a particular concentration of gradient of molecule change, or also if they change trajectories in response to this particular molecule, and how this particular molecule distributes within our system. So we can see all these points later during the practical demonstration, which this will be a demonstration, but we can develop after maybe in the afternoon or this evening. And this will be for that the last cell imaging software, which is really that you may know, and with that we do some to analyze this kind of parameters. So I hope you are telling about it. So summary, yes, 3 minutes. During this class, we have seen how basically cell migration mechanisms are important for the good functioning of important biological properties. We have seen and we have considered how it is important when we are in a micrometer scale to take into account other physical constraints, such as the viscosity of the liquid that normally we can take into account in our conditions. And we have seen how microorganisms were able to develop propulsion mechanisms that enabled them to overcome and move in this difficult condition, I would say. We also have seen a little bit how works the mechanism of sensing in our prokaryotes or eukaryotes, small cell or mid-sized cell. And a little bit of focus on the Phytophthora, which is a zoosporic telluric pathogen, and on the zigzag model of plants. So a little bit of plant immune concept. And finally, how microfluidic can be an efficient tool for the analysis of the function of zoosporic pathogen, but also of other swimming cells. So I thank you for your attention, and I also would like to thank my team, ParAqua team, and also the team from the Institute of Nice that I'm working with them, and we want them to collaborate. And that's all. Thank you.